let me unveil this mysterious title. I'll be talking to you today about the smallest self-replicating bacterial cell. And unlike Camila or Anya yesterday, I did not bring any to show you and spread around, except the bacteria that I have on me normally. So let's start with, I would like to tell you first that there is a long-lasting trend in technology industry. It's a trend for miniaturizations. We have smaller and smaller cell phones, smaller and smaller computers. And this trend was also brought to biotechnology, but about that in a second. First, a question to you guys to warm you up. How many genes do you know? Or if you're not, how many do you think we humans have? So can you tell me by show of hands, how many of you think it's A, 2,000? Okay, B, 20,000? Yeah, and C, 100,000 genes. Okay, so the right answer is around 20,000. So you can be proud of yourselves, but this is something that scientists fight about. So it's from 20 to 25, and that's even a rough number that they cannot agree on. So now to come back to miniaturization and put that number into context. In 2016, some scientists from California created the smallest bacterial cell, which has genes only essential to survive. It also replicates by itself, duplicates in 3 hours, which is a reasonable time, and it grows in a nutrient-rich dish. So if you imagine these are minimal cells, and they grow in an environment full of nutrients, such as amino acids, vitamins, and nucleotides, and these nutrients are absorbed into the cell and used for the cell to perform its biological processes. So again, only genes essential to survive and to fulfill these 3 conditions. How many genes do you think that is? How many genes does this minimal cell have, roughly? Maybe anyone can guess? Okay, I'll tell you. It's 473 genes. So this is 42 times less than we humans have. So only 473 genes are necessary to sustain life. But when they created this cell, they didn't know functions of 149 of them. So they didn't understand 1/3 of essential processes that sustain life. So as you can guess, our aim was to try to identify these missing essential functions using computational approaches. And to do that, we used sequences of proteins, which are products of the genes. And you can see here an example of a sequence. This is a sequence of amino acids that the protein is built from. And me as a computational biologist, I do not wear a fancy white lab coat. I just work with a computer and files, many, many files, day by day. So just a little bit also about proteins. A relationship between a gene and a protein is like you can imagine a relationship between specifications and machines that are created based on those specifications. So in a cell, gene is read, decoded, and then translated into a protein. And expanding on that analogy, proteins function in a cell just like machines function in a factory. So they have different roles. You might have some machines that fold the boxes. You might have some machines that put stuff into the boxes and then that close the boxes and ship them off. And I think the most well-known examples of proteins is hemoglobin that transports oxygen around the body, and it lives in red blood cells. And my favorite example is a kinesin that transports large cargo, for example, other proteins. And if you love biological animations as much as I do, then you Google a day in the life of a model protein, and you're in for a treat. So because experiments to identify functions of genes are very time-consuming and very expensive, computational biologists constantly look for new, better methods to predict protein function using sequences of amino acids. So we basically can distinguish two main approaches. Either it's a similarity to other proteins. So if you imagine that these four shapes are proteins, then if we know a function of a rectangle, we can assume that the function of the square will be similar because they are similar. And if we can't find any proteins that are similar to our proteins, we can also apply physical and chemical properties of those proteins. For example, composition of amino acids or location in the cell. Like, for example, here, if the square lives in the nucleus of the cell, but the rectangle lives in the membrane, they might not have so similar function after all. So we used plenty of methods that predicted different protein properties. We predicted similar proteins which are evolutionary close, so they come from the same ancestor. We predicted three-dimensional structure of the proteins when we could. That wasn't always possible. We predicted location in the cell, small molecules that bind to the protein, and gene ontology terms, which are basically terms that describe the function of the protein in a very standardized way. So here are our results. We predicted more informative functions for 66 out of these 149 proteins. And as you can see here on this beheaded by data visualization specialist pie chart, 50% of genome, so we combined the results that we got and the results that were initially provided. And we saw that 50% of this minimal genome is responsible for reading and expressing of genetic information and for preserving the genetic information. So basically to preserve the genetic material and to pass it onwards. But we also identified 24 new transporters. So to show you an example of a transporter, this is a protein. We predicted the structure of it. And this protein lives in a membrane of the cell. And we predicted it to be in the membrane. We predicted it to be a transporter. But we do not know exactly what it transports. So it might transport some nutrients from outside of the cell inside, like a vitamin here. Or it can export some metabolites from inside of the cell outside. So lastly, we came up with a conclusion that there is no one minimal genome. But minimal genome depends on the environment it grows in. So each minimal genome has essential set of genes that is a prerequisite for all the bacteria. And then they also have additional sets of genes that enables life in a particular environment. So, for example, here you have 3 different dishes that represent 3 different environments. And the first one is has the highest number of nutrients, and the last one the lowest number of nutrients. And the number of genes that are responsible for metabolism and biosynthesis will be highest in this environment, but lowest in the first environment. And the number of genes responsible for transport will be the highest in the first cell and the lowest in the third, because here all the nutrients are provided, so the cell can just import them inside, but here there are not a lot of nutrients, so it has to create them. It has to first degrade existing proteins, for example, and then synthesize the nutrients. And thank you so much for your attention. I think I spoke very fast and for like 3 minutes only. I would like to thank my supervisors, Mark and Martin, and a bunch of people who made me create this presentation. And they were very patient with me and many attempts of translating my research into a popular science talk, which was much harder than I assumed initially. So thank you, and if you have any questions, you're welcome.